Hello, EEB. Welcome back to the grand finale of fall semester Tuesday lunch seminars. Before we begin our introduction, fourth floor would like to announce that last night, late at night, somebody delivered a ping pong table into our little atrium area. And so if anybody wants to come join the competition before they take it away from us, please. It's like brand new. Yeah, it is from the Edward Henry Krauss ping pong table is part of the Great Take a Break tradition. So basically there's a group that buys ping pong tables and delivers them into random buildings on this campus, and fourth floor was blessed last night. I don't think it will last, but you gotta come play. Okie doke. But before we play ping pong, we get to hear even more fun than ping pong. So welcome to our last Tuesday lunch seminar of the semester. It is my pleasure to introduce Dr. Erick Bayala, who is currently an MCDB research fellow in the Wittkopp lab. Today we'll be hearing about Erick's incredible Ph.D. work and hopefully seeing many of his stunning photos and animations. Please join me in welcoming Erick for his talk, How to Paint a Butterfly, Developmental Insights into Heliconius Embryology, Color Patterning, and Pigmentation.
Let me clip this somewhere. Hello? Do you hear me?
Yep.
Good. So let me start a timer here so I know that I'm not over. Thank you everybody for being here, and thank you for the organizers for putting this together. And as Emma was saying, I'm going to be talking a little bit more about my PhD work. I hope to also transition towards the end of what I'm doing here. So it's going to be a little bit of both my butterfly research and my current fly work. So the title of my talk is How to Paint a Butterfly, Developmental Insights into Heliconius Embryology, Color Patterning, and Pigmentation. But before we go there, I want to tell you a little bit about me and how I got to where I am and how I got interested into this. So my background is a little bit unique. I came from an art background. I did an art specialized high school and also did a dual degree during my undergraduate in both microbiology and visual arts. So very early on, they asked me to choose one or the other. And I think that my PhD and my future work has kind of been this merge of both my art side and my scientific side. So I developed very quickly into like scientific illustration. And from there, I fell in love with microscopy. So a lot of what you're going to see today is my passion for both how shapes are created in nature and how can I represent or show the science we do with them. So I didn't want to include this as the first slide, but it's the second one. So this is the typical diversity slide. And I don't have to tell you that the world is very diverse, but the kind of diversity that I'm very fascinated for is color pattern diversity, especially how these shapes are created when you think about the developmental process of how you make this. So we start all from an on-pattern single cell starter point, and then we end up in a functional organism that also has to have color patterns associated with this process. So we go through this developmental process where you have an intermediate embryonic state, and then you finish in a fully formed organism. And here are some examples of animals that have been used for pigmentation studies. Not the peacock, that's probably just a glorified chicken. But the main thing that I wanted to get is that we have approached this pigmentation question from different systems and have learned how form and shapes are created through developmental processes. I specifically can put this in an umbrella question of how are color patterns built during development. And specifically, I try to target this question during my PhD work with Heliconius butterflies. And Heliconius butterflies within the Heliconius group, you can find that they're pretty diverse among themselves, but even within this diversity, you can start teasing apart specific components of the color pattern. For example, you can see the red color patterns, melanic color patterns, which are black, then you have white variation and yellow variation. And all this variation that you see here is a mix and match between all these four color components. And when you think about it, you can think about it in distinct elements. For example, black coloration or melanic coloration is controlled by this gene called Wnt. And later on was discovered that also another gene called cortex also controlled this melanic variation. Then if you go to reds, you have optics, which is the gene that controls red pigmentation. And when we got to white and yellow variation, we didn't exactly knew how genetically the variation is encoded. The other cool thing about it and how I got fascinated about this is, if you notice about the genes, they're pretty unique kinds of proteins that are coming out of them. So we have a signaling ligand with Wnt, we have a cell cycle regulator for cortex, and then we have transcription factors in the case of optics. So this already is very interesting developmentally, because if all of these things are putting patterns together, they're doing so in very different cellular ways. And white and yellow, through research in the Cromford's lab, which is the lab that I was part of, was identified to be controlled by this gene called aristaless. So most of the talk will focus into aristaless in just a second, but I just wanted to give you that both pigmentation can be diverse, but also can be very simplified when you think about just the genetic elements that are controlling this process. So before we go specifically into what I did during my PhD, I wanted to provide both a developmental context and a cellular context, because I think it's very important to be able to position the kind of work that I did. So we have here the typical development process of a butterfly, although one of the things that I, again, from my imaging and microscopy background, that I did was to expand what we know from butterfly embryology. So this is the embryological process of a butterfly, made on schematics. And then we have 5 larvation stages, so 5 instar stages for larvae. Then we have pupation and eventual eclosion of the butterfly. And a few things that I want to say here already is that the wings, which are the functional units that are becoming pigmented, are already developing during this caterpillar stages. So if you go grab one of these caterpillars and cut them through the side, you can actually see the wings already popping out of them, which is something that is kind of crazy, and I didn't even knew when I started working with this. So now to think about the cellular context that we're approaching, so I told you just the development time. We can also think about the specific cells that are the ones that are adopting a specific color fades, and these are called scales. And the scales, you can think about them as pointillism. They're just only grabbing a single color fade, and the color pattern that we see is just a combination of all these scales together. And what we can see here is that development process through the development of the butterfly, where we have this magenta cell, which is called the scale building cell, and this cell starts growing as time continues. And then there's this dark blue cell, which is called the socket cell, and just keep it in place. And as these cells grow, when you see that it becomes gray like this, its cytoplasm collapses and it dies. And this skeleton of the scale is what becomes pigmented and what we see in a butterfly at the end. And then we can think about the genetic control that underlies this process, where we have the same genes that I was just telling you about, cortex and Wnt, which happens very early in larval stages. They're doing their job over there. And then you have other genes, which is optics, that does this during pupation. And then at the end of the day, we can test this by just looking at the expression here. You're looking at the expression here in purple from other people's work. And the kind of work that we do is we look at whether the expression pattern of these genes resemble the future patterns. So for example, with optics, it's very clear that the place where optic is expressed here in green resembles especially where the red parts of the wing are going to be. So the other layer that I want to introduce is that I call this entire section the patterning phase, where you're creating pretty much a blueprint of the entire system, while this black box over here is the realization phase, where you're grabbing that blueprint and now grabbing all that positional information and making actual pigments. So these are the last enzymes that are all working together to put together the final color patterns. So where does white and yellow fit in this entire thing? And that was kind of like my PhD work, trying to figure when in this blue box does aristaless work, and also how does it relate with this realization box that I have over there. And I am going to be talking especially about aristaless 1, and why that one is there is going to become obvious in just a second. So one thing that we did just as a proof of concept that we are in the right way. So aristaless was knockout, aristaless 1, and when this knockout was done in butterflies, it's a mosaic knockout because we cannot really target every single cell in the egg. And you can see that this is a genetically white butterfly that has clones that have turned from white to yellow, showing that aristaless 1 is very important for being a white butterfly, and if you break it, you become a yellow butterfly. And the other thing I want to clarify why white and yellow is important and why keeping them together is relevant is because white is the state where you don't have any pigment, while yellow is just a state where you have a white scale that just happened to grab this yellow pigment before you eclose. And that's very critical because it happens 20 to 30 minutes before the butterfly ecloses from the pupae. Okay? So, aristaless, and I want to just take a second to talk about why I say sometimes aristaless or aristaless 1. So aristaless, from Flyware, we know that it's classified as a parent-like domain transcription factor, and it's very well known for being important for developmental roles, but it's especially known for its role in appendage formation. So everything that is sticking out of the body of an insect or some animals is usually controlled by aristaless activity. And it was already named based on the phenotype of the missing arista, so this little hair that the flies have coming out of the antennae. So when we go to a few animals, we can see that anything, like I was saying, that sticks out of the body, that has a little bud that comes out of the embryo, is highly antigenic for the activity of aristaless. So you will see a lot of aristaless activity in those tips of those appendages when they're coming out of the body. And this is the case for many, many more examples, but I'm just giving you here a few. So what about Lepidoptera? So in Lepidoptera, there was a really interesting case where aristaless was duplicated, and this duplication event happened at the base of Lepidoptera, so both butterflies and moths have it. And the cool thing is that one of the first things that people were searching whether one of the aristaless or both of them have this developmental function related with appendage. Although embryology as a science in butterfly doesn't really existed until a lot of my Ph.D. work, the biggest question was, what system can we study that has an appendage, right? So they started with moth, and you can see here that this is your Bombyx moth, normal antennae, and what happens when you use morpholino, which knocks down the activity of aristaless. You can notice that a lot of these branching patterns are messed up. And also they notice that it is expressed at the tip of appendages, similar to how you would expect from the unduplicated version of aristaless. And this is the current model that they were thinking, that you just press it in this little tube that allows these branches to grow. So that's what we know from aristaless. And I wanted to now put specific questions that I'm going to try to answer a little bit. And one of the first things that I did in my PhD was trying to figure out whether aristaless 1 still have this appendage role, but in the embryo. Whether the same things we see in gorillas or any other insect, do they hold water when we think about aristaless 1? Then I shifted to wings and butterfly pigmentation to ask when in the development process does aristaless activity work, and how that activity leads to white and yellow decisions. So how ultimately these genes make you white or make you yellow. And then the question that you might have already in the back of your head, how do we go from something that does patterning of appendages to something that does pigmentation? What is the link between these two things? So both of these papers talk a little bit about this, but I'm going to mostly focus on the first one. But if you want more information, you can see, you can take a look on both of them. So for the sake of time, I'm just going to grab a single time point today and just take it out of the egg and then show you a little bit of what this imaging looks like. So what you can see here is one of those embryos where aristaless is here in green. So this is targeting the protein. So this is an antibody that targets specifically aristaless 1. And you can already notice, without me telling you anything, that we were looking for aristaless 1 at the tip of appendages, and we saw it, right? There are a few things that are unexpected. For example, this dorsal furrow that you can see here, that has not been described in the literature as a thing that aristaless does. But what's very interesting and very consistent as well with our embryos, and the other part that you can see is, so when we think about appendage, sometimes we only think about legs and things like that. But for example, the mandibles are also appendages, and the eyes are sensory appendages as well. So you can see this little demarcation of the future eyes as well as having aristaless 1 activity, which is pretty cool. So the cool thing that we can do here is that we can grab these embryos and do 3D reconstructions just to really get at whether the internal parts of them also have any aristaless. We wouldn't expect that, but what I did is just I went took images through the entire embryos. These are very transparent embryos at this stage, and that lets us know whether exactly we are looking just at the surface level or whether there's anything inside that also has aristaless. And as you can see, it's mostly related with these appendages and this dorsal part of the embryo. I love these spinning embryos. So looking specifically into the structures that I was telling you about, mandibles, legs, so this will be the thoracic legs and abdominal legs, you can notice that aristaless at the tip. The other part I want you to notice is where nuclei are with respect to where aristaless 1 is. This is a homeodomain transcription factor, so you would expect it to be nuclear, right? If it's doing its activity. But at least during any of these images, I never found aristaless 1 to be collinear with nuclei. And just to get that point home, I will show you in a second even closer views of it, just to get that we never saw aristaless 1 to be collinear with nuclei, at least for these developmental stages. So one thing that we did, we have our CRISPR setup very well put together for our wings. So I went ahead and grabbed our CRISPR setup and used it to target, to knock out aristaless in embryos. And this is one of those embryos where almost half or a quarter of the embryo is knocked out. So this will be your normal appendages extending. And when we take it out, especially down here, the entire body breaks down. But a big part of it, especially if you look between these two ones, you can see how these appendages are heavily affected by extension. We know for a fact that if we do these knockouts, these butterflies survive. So there has to be something else that helps them become a normal butterfly. But although I won't be showing this today, if we do RNAi to specific stages of it, we can also affect the adults. Like we can do it during pupation, and we end up with butterflies that are missing their claws, they're missing their proboscis. So something about the system still is able to produce a fully functional organism, but depending on when you knock it out or knock down this gene, you will going to have different defects on these appendages. So far what I have told you is where aristaless is expressed, at least in embryos. So we have confirmed whether aristaless is expressed in the right place at the right time. And the other part, that if we break down, if we knock out aristaless 1, we do see developmental defects at the level of appendages, meaning that it has a level of functionality with respect to appendage formation. So the other part that I was telling you about is whether this location with nuclei ever happens. So here you're looking at a seaplane, you're looking through the body, and you're mostly looking at the abdominal legs. And the main reason why I'm showing you this movie is so you can see where aristaless 1 is detected versus where the nuclei are. And as you can see, there is not an overlap between. And the reason we have to do this is because this tissue is very thin. There's only a couple of cells at these legs. So it's very tricky to infer whether aristaless is overlapping with nuclei or not. So it's important to go to this level of imaging. So we can reconstruct this by doing a maximum intensity projection just to show how aristaless is distributed in cells. Okay, so I just told you that aristaless 1 is expressed on these appendages, and if we take it out, we start affecting the development process of these appendages. But what about wings? So just to remind you, the functional units for wings are these scales. And I will tell you actually now about a lie that I put at the start. The lie was that I told you that when we knock out aristaless, we get white parts of the wing and make them yellow. And it's not 100% of a lie, but if you might have noticed that there is not the only change that happened. So one of the first things that I did in the Comforts lab when I joined was to notice these clones that were not described before when we knock out aristaless. So we not only change the white parts of the wing to yellow, but we also are modifying the melanic parts of the wing to brown. And this was very interesting because we were just thinking that this was a gene that is controlling a switch between being white or yellow, but this already is telling us that something else must be happening in this system developmentally. The other cool part is that this brown that is created here is not present in any other part of the wing. So this is not that we're changing the scales to another state that can happen on the wing. This brown is not present in the entire wing, meaning that whatever it is not the typical developmental trajectory for a different pigmentation outcome. So The other part that we did is we checked whether yellows also have an effect there, because if I told you that aristaless 1 is important for white, if we knock them in yellow, they shouldn't have an effect in this part, but we still see the effect on the melanic parts of the wing. Okay? So how does aristaless look in a wing? As you might already be expecting, if I just told you that aristaless 1 is important for the entire wing, we just noticed that aristaless 1 was pretty much everywhere. And this, from a patterning sense, was kind of a little bit disappointing because you were expecting something that is just in a pattern. But I promise it's coming. So the main thing is that aristaless 1 was everywhere. And when I say everywhere, everywhere. So we can see it distributed across the entire wing. And then when we look at the individual scales that are making these pigmentation decisions, again, you're looking through the tissue into the nuclei of these cells and then coming out from the other side of the wing. You can see how the entire scale is loaded with aristaless, except for one part, the nuclei, right? And that's very interesting considering what we knew from embryos, right? So just to, I will play that in a second, but just so you can see a 3D reconstruction of this. And these are the scales, and you can see in green is where aristaless is. Red here is actin, so these scales are actin-based before they die and become chitinized. Here you can also see the nuclei. The big nuclei are these scale-building cells, and the smaller nuclei that is next to it is the socket cell, the cell that is holding it together, right? And I don't need to tell you that, yeah, we couldn't show any moment where aristaless 1 is within the nuclei, which again is important considering this is a homeodomain transcription factor. And again, just to show you the video, now that you know what you're looking at, you can notice how the entire scale body is just loaded with this aristaless 1 protein. And then when you go to this nuclear layer, you're pretty much like removing aristaless 1 from those nuclei. Okay, so, spoilers. So what about yellow? So one thing that we noticed in yellow was that there was this very narrow window of development where aristaless 1 just goes down in the middle of the wing. So there's a reduction that happens only for an hour to 2 hours, considering that these are 8-day windows of time for the entire pupil development. It was not as easy to find this developmental window, but the main thing is that we see how this happened. And the other cool part that you might notice is how this triangle is shaped over here is kind of different to how the triangle shape is done down here. And it's because underlying from this bottom part, you have what is called the sex band of the butterfly, and that part is heavily yellow pigmented, which is why we're also thinking that this part is also lacking aristaless at that same time. So can we test whether this absence of aristaless during this specific time point lead to the pigmentation difference we see in our CRISPR clones? So we went ahead and grabbed some of our CRISPR wings and tried to find places where normally you will have aristaless, so this is a white butterfly, and see what happens in a CRISPR clone. And here we can see a part that definitely should have aristaless, and especially the shape of the clone is pretty unique. And you can notice how this entire region is now lacking aristaless 1. So far I have told you how do we start to make this decision happen, but I haven't really gone at what is happening in the middle between this difference in aristaless to then having a pigmentation difference. And I feel that this part is very important because so far, one thing that is common in the field of butterfly developmental biology is this, is finding how a pattern resembles the future adult pattern, but a lot of times we don't go further and we don't ask how this difference in pigmentation expression pattern leads to difference in pigmentation. So I went ahead and had this model and I wanted to check what is happening in the middle. What is happening when you have aristaless and when you don't have aristaless for a yellow. So this is more or less what we know from other systems of how yellow pigmentation comes into the pigment granules that are in this scale. We have transporters as a way of carmesine, white, and scarlet that are moving in either the yellow pigment itself or a precursor. And then we also have enzymes that are needed to turn the precursor into this yellow pigment. So one thing that I did was I wanted to check whether any of these components that are needed for moving or synthesizing yellow are upregulated or downregulated following this activity of aristaless 1. Okay? So what I did, I grabbed wings for both white and yellow and cut them into pieces, cut them into 3 different pieces as best as I could using the veins pattern, and then ask how is the gene expression different between being white or being yellow. And just to orient yourself a little bit of what the data looks like, you will have 3 different time points, which are down here. You have also relative expression over here, and then you have a line for white or a line for yellow, and then you have a set of 3 graphs per specific enzyme or transporter. So for the first one, we noticed that they were pretty much the same. But then for the second one, we started noticing a pattern where around 6 days, which is when these scales die and they flatten out, where a lot of these enzymes that are putting pigments into the scale body are active, we started noticing this pattern, and the pattern became more obvious as we started adding more and more genes into the mix, especially for the transporters for these 3-OHK. Although our p-values are not the greatest, so yellow butterflies are really hard to find, especially here in the US. We did the best way we have, but it was, once you add the 3 different transporters, it was very obvious that yellow wings were upregulating all these transporters. They were upregulating the yellow transporter and also that one enzyme. So far, one thing that we noticed that was in the middle part was also happening a little bit to a certain degree in both of the other parts. And the explanation that we have for this is mostly due with the manipulation that we were doing. It's really hard to consistently cut the band of the wing out. And even when you do with the vein pattern as a landmark, you have to cut some parts of the white or yellow tissue into your both sides, given that these lines are not straight, right? So I feel like the reason why we have these effects over here is because we're grabbing a little bit of these white and yellow cells into the mix with the melanic cells. But it will be a good thing to follow up and just grabbing, for example, closer to the tip of both sides and then see if that solves this. So now we have a little bit of a bigger model. We have that the lack of aristaless activity here leads to an upregulation of cinnabar and 3 transporters, and these transporter enzymes are needed for yellow pigmentation. And one thing that I wanted to take it further for my PhD work is I wanted to test whether the upstream signals that are normally important for appendage formation were also there for wing development. And although I won't have that much time to show this today, I just wanted to let you know that we did test this, and we find out that Wnt signaling, which is one of the main activators of aristaless activity in appendages, is also involved with activating aristaless across the entire wing. And I just told you at the beginning that Wnt is one of those signals that really patterns one or the other. So you can already think about it as like Wnt activity, which is established very early in larvation, is already setting up a cascade of events that would eventually lead you to having white or yellow pigmentation patterns. Okay, so what I told you so far is that aristaless 1 is upregulated in white pupil wings. Its expression is reduced in the yellow ones. Aristaless 1 knockouts, which produce white to yellow switch, also shows those effects in expression. And finally, that after the reduction of aristaless 1, there is an upregulation of specific enzymes and a set of transporters that are needed for producing this yellow pigmentation. Okay, so the last part that I wanted to tell you a little bit about is how does these 2 things link together, and also get a little bit towards this why we never see it in the nucleus, which is, I feel, a very interesting feature. So I think a big function of aristaless 1 activity is rooted in what is called the heterochrony hypothesis. This hypothesis for butterfly development postulates that different fates of scale coloration are controlled by when they are ready to achieve this input of specific pigments. So in this case, where it was discovered, they discovered that yellow scales develop quicker in this system than melanic scales. And actually, by shifting this time of development without changing any of the gene expression, you can change the outcome of this developmental trajectory for these scales. So given that aristaless is just kind of like a glorified appendage for wings, how does that fit within this? And I think that a big part of like how aristaless work with making things extend or not tends to affect how ready they are to accept pigmentation. And this is very similar to how we know, for example, pigmentation works in mammals, where we already have information that certain hair follicles are ready to be able to accept all these enzymes that are needed for melanic pigmentation or yellow pigmentation, while other ones, if they're lagging behind, they miss these windows of development where the enzymes that are present are needed for then those pigments to be achieved. And that's how, for example, difference in the striping patterns are achieved in rodents. Similarly, and I just want to point a few examples. Again, this could be completely, and this is where I run out of PhD time. One of the things that I have started noticing is that often these systems where you have differential time on development for the cells that are becoming pigmented or not are often controlled by ALX. ALX stands for aristaless like homeobox. So this seems that aristaless, at least across different systems, has been implicated with pigmentation decisions, but more importantly, has done so by altering when these pigmented cells or these pigmented units are ready to achieve the level of enzymatic pigmentation needed. So what about my system? Do I have any evidence in favor or against this? So the only piece of evidence that I got towards the end of my PhD, which I think will be really interesting to follow up on, is the amount of actin that is created when you are in an aristalin present part versus an aristalin deficient part of the wing. So this is the amount of actin that you will normally see whenever aristaless is there and when aristaless is not there, right? So one thing that we found here is that if you have aristaless, meaning you're white, you have a lot of this lack of actin, meaning that you're, in theory, developing slower. While if you're yellow, you have a lot more actin, meaning that you're developing faster, which means that this allows your scales to be ready in a certain way for accepting this yellow pigment that just happens 20 to 30 minutes before pigmentation, before eclosion happens. And when I say is a few minutes, so one thing that I tried to do towards the end of my PhD is to image when yellow was coming here into the wing. And again, it happens so close to eclosion that when I started seeing yellow come in, the butterfly came out of the chrysalis on me. So I got a really nice alert on my scope saying something is hitting your objective. And it was the butterfly saying I'm here. So that's the only piece of evidence that I have of that. But one thing that I will say that could be done in the future is to really play with the development time and leaving aristaless intact, just to see if you can really modulate this. The other thing that I did is I changed wing signaling, sending scales later, sending them back in development. So I have wings that have both smaller scales and bigger scales, and those scales already show difference in aristaless amount. So it could be related or indirect, but at the same time, it could be a very interesting thing to follow up. So I told you a lot about aristaless 1. I need to tell you also a little about the neglected cousin of the family. So towards the end of my PhD, I wanted to search whether I can get anything into the nucleus, because at that moment I was doubting myself. And one thing that was really surprising and cool was that when we image aristaless 2 in the same time points, we can see how aristaless 2 is in the appendages, but it's very nuclear, right? So this is not that I am unable to say anything that is inside the nucleus. As you can see, it's very personal now. But you can see that aristaless 2 does go into the nucleus at the same time. The other part that was very interesting was that although we cannot necessarily work in aristaless 2 for, I will tell you in a second why, in adults, we can work on it on larvae. And so these are the larvae of Heliconius, and you can see they have these pigmented spines, which are pigmented conveniently. And one thing that I did is to check whether aristaless 1 or aristaless 2 are expressed in these spines, and then I saw a lot of aristaless 2, but no aristaless 1. And that was very interesting, given that I just told you that aristaless 1 is always in the appendages. So I went and dig a little deeper. First, check whether something was lying to me. I did in situ. It also shows that it's on the spines. And then I did also CRISPR knockouts of aristaless 2, showing that these spines are not only malformed, they're also lacking pigmentation. So whatever aristaless 2 is doing is also related with pigmentation. And you might say, why you haven't done it in butterflies? And the reason is because these CRISPRs are extremely sick and they die very quickly after pupation. So whatever aristaless 2 is doing doesn't permit the after pupation to happen. So I went ahead and grabbed these spines and looked through a developmental time series for them. And then something very clear came out. So here you can see these little stubby spines coming out of the epidermis. And then as you can see, the spines are getting bigger. But one thing is disappearing, it's aristaless 1. While aristaless 2 starts very lowly expressed, and then it ramps up onto this everywhere, right? So I noticed that there was this developmental discontinuity between aristaless 1 activity and aristaless 2. And like I was telling you, aristaless 2 is localized nuclearly, which might be a very interesting point of follow-up in the future, finding what is the relationship between the two of them for the function of the genes. The other really nice piece of evidence that I have here is the specific cells that are more that have more of aristaless 2 are these cells over here. So there's 2 nuclei, one of them is neuronal and the other one is structural. This is the one that puts together the spine but also makes it pigmented. So you can see how there's already a difference between those 2 nuclei, hinting towards aristaless 2 having activity with respect to pigmentation. Finally, another pigmented unit or functional unit that I looked at was eyes, just to see whether my spine's developmental inference holds up when I go to eyes. So this is early in development, aristaless is there, not in the nuclei, as expected. But then when we look later in development, we can see how aristaless 1 is not there, but aristaless 2 is. Furthermore, if we look at these pigmented columns that are underneath the simple eye of a caterpillar, you can see how aristaless 2 presence perfectly correlates with those pigmented spots that you have under the eye. So this led me to this other model that I discussed in my second paper that talks about how you have this continuity between aristaless 1 to aristaless 2, aristaless 1 being everywhere but in the nuclei, and that leading to aristaless 2 being in the nuclei and eventually everywhere. And something is very sus about aristaless 1. Some of you might understand this one, other ones no. So from there, I just wanted to wrap up this part by saying we have, and we think that the heterochrony hypothesis is the key to understanding how functionally aristaless activity and in appendages and wings is linked together. And also, aristaless 2 does localize to the nuclei, thank God. And so I'm able to also notice this difference in temporal and cellular localization between the 2 aristaless, which is very interesting. So on the few minutes that I have left, I just wanted to tell you what I'm doing currently. And the big part of it still holds the same in terms of I'm still interested in how patterns are created, especially looking at how or where do you tweak things in order for patterns to change. And that's a big difference, I would say, from my PhD work to my current work. I was very interested in knowing how a pattern is put in place in the first place, but now that I know a lot more about what are the elements needed to the pattern, I want to see how you modulate these elements in order to create pigmentation diversity, right? The good thing about fly pigmentation is that a lot of the components of the fly pigmentation enzymatic box, right, this realization box that I told you at the start, are well known. However, this regulatory control is like this Christmas that keeps on giving. It's just every single day we discover more and more elements that control these enzymes that are doing the terminal pigmentation stages. So one thing that I did, I developed and pioneered a technique called HCR for these very troublesome tissues across non-melanogaster flies that allows us to investigate what is different between very closely related flies that do have difference in pigmentation. So we have some of them that have these melanic spots on their wings, other ones that have difference in melanization in the thorax or in the abdomen, and same with the leg. And due to like some genetic mapping that we have, we know whether this difference sits down in the regulatory box or whether this difference sits down within the realization box. So one thing that I'm doing is I'm using this technique called HCR that allows me to just see all these genes at the same time and adding more and more genes into this process in order to try to answer what is changing, how is it changing, and also knowing about how long are these windows. That's one thing that we have been misled for many years about how short some of these developmental windows are for the expression of these genes. And the reason I say we have been misled is because a lot of time when we work with reporter assays or protein assays, we lose a little bit of where expression is modulated. So one example of this, for example, of what I have done this year, you can see melanogaster where you have normal banding, very thin band. But then when we look at the one that has thicker melanic band, we can see how the yellow expression just goes across the entire band. Or we can go also to see whether this yellow expression, as you would expect, is there on a developing wing. But the cool thing that I already have noticed is that this expression window of yellow is only like a couple of hours compared to like the time the protein just lasts there. Okay, so thank you to everybody that has been important for this work to happen, especially like to the two postdocs that were key for my PhD work, and also my student, and also for the Wittkopp lab to being like a new home for me. I will answer any questions. Yes?
So this aristaless
Yeah.
one, you said that it's a homeodomain transcription factor. So do you know any other examples of genes that are transcription factor?
Yep.
But they also do have other functions, and you can find them outside of the nucleus.
Yeah, so that's one of the things that we wrote a lot about, because one of the biggest criticism we have is that this is a homeodomain transcription factor should be in the nucleus. And as you can tell from my talk, that takes it very personal. But the main thing is that yes, there's other homeodomain transcription factors that do have their role outside of nucleus. And that can be related with dimerizing with other transcription factors sometimes to just keep things out of the nucleus. And that's the first thing we were thinking about, given that aristaless 2 does go into the nucleus. You can think that maybe it's interacting directly with aristaless, but it could be interacting with the thing that upregulates aristaless 2, right? If I'm just keeping you hostage outside, it's not until my aristaless 1 expression goes down that my hostage scenario can leave, then that thing can go into the nucleus and then drive aristaless 2. So that's one way to think about it. Other ways is that these transcription factors sometimes also have like phosphorylation capabilities that allows them to be active outside of the nucleus. Especially for the first scenario, so aristaless 1 does have a dimerization domain. And it has been shown, at least in flies, that aristaless homeodimers happen, and also heterodimers with another protein called clawless. So there's evidence for aristaless activity being associated with dimerizing with something. Yep.
I was curious, do you know, is anything known about the adaptive significance of the melanization in the caterpillar spines?
Yep. So at least in butterflies, I don't know that much if this holds up for caterpillars. I think it does. So wound healing in butterflies is associated with a melanization trajectory. And what happens is that when you melanize something, it becomes very rigid. So if you poke a hole into a pupae, which I have done plenty of, you will see that the next day, there's kind of like this scalp version of it will be just a melanized blob on top of it. And I feel like with your spines, a lot of the time you want that. You want an extra level of rigidity. So then you don't like get the bend. But then I think your question is also interesting from the direction of what came first, right? Like if I knock out aristaless 2, am I seeing these spines bending because I don't have pigmentation, or does my aristaless 2 effect is affecting both of the things? And so far we don't have the answer. And like I said, when we do aristaless 2 CRISPR, most of the things die. So it's very limited amounts of data that we have from that.
That was really cool. I was wondering, so Heliconius is obviously studied a lot for mimicry.
Yeah.
And I know that there was like one paper that showed that the way the model species tells itself apart from mimics is because of the 3-OHK pigmentation.
Yep.
But do you think that in terms of mimicry, is it at the level of expression of aristaless 1, or is it at a higher level in terms, do you think that the mimicry then happens at the pigmentation level?
Oh, that's such a good question. And I didn't work on this particularly when I was in the professor, but a big contingent point was very similar to what you're saying, is that the mate selection gene, or like the locus that makes you select white for white or yellow for yellow, like they select on specifics, is also in the same place where aristaless is. Right? For a lot of time, they were thinking, is it aristaless doing both things? Is this the gene that does pigmentation, also the gene that does behavior, also the gene that does appendage in the right place? And so far, they believe the peak is a little bit shifted, and they have landed on another gene that is very close to aristaless called senseless, and that one controls eye development. And the reason that matters for TRIO-HK is because TRIO-HK is this pigment that serves a lot of functions and one of the other functions that it does, it coats the eye filtering pigments, which I think might be more related with that role of TRIO-HK in mimicry. That said, I'm not 100% convinced that aristaless is out of the question or out of the contingency for this behavior trait as well, given that I have already shown that it's expressed in the eyes as well. So maybe one thing is related with the other.
So just as a follow-up though, so in the knockouts that you showed, for example, there was more of white, and then there was also loss of melanism in the black parts of the wing.
Yep.
So in terms of the mimics though, do you think that the expression of this gene itself regulates the amount of pigment that's going to go into the wing?
So that's a good question. I don't know if it regulates the amount, but more about the decision, right? Like I think aristaless specifically is telling you whether you are ready or not to become a yellow scale, right? And the reason why I say ready or not is because I didn't show it here, but I did a lot of experiments where I modulated aristaless, not completely take it off by using drugs. I drugged my butterflies. And when I did this a little bit of experiments, I noticed that the decisions were always binary. They were not more yellow or more white or more black or more brown. They were just either brown, black, yellow, or white, right? So I feel the model where scales adopt a single fate still holds. That said, on the mimicry side of things, one thing that the lab has been following up is testing these butterflies for mate choice trial, right? Whether there's any evidence for some underlying capabilities of these scales to be attracting females or males differently. I don't have exactly what is the end result of that. I just know that CRISPR butterflies, they just do worst in mating trials. But that could be just because they're missing limbs. Or yeah.
So as somebody who thinks a lot about like color patterning, I do not. And I have like recently come across the fact that like there are UV patternings and patterns outside the spectrum of visible light that like, could play a big role in mate choice and things like that. So do you spend a lot of time thinking about that? Are the methods similar? Yeah,
no, it's such a good question in terms of, so when I started my PhD, I was thinking like I could do everything, like we often do in PhD starting scenarios. And it was very clear that I couldn't do that part because these white scales, they're not just white, they're also extremely UV reflectant. And when they become yellow, they lose that UV reflectance. So this aristaless role is also related with how reflectant or not reflectant these scales are, which affects how the mate perceives them. But again, because I was already doing all of this, I couldn't follow on that. But the lab did a lot of, you put the butterflies into solutions that takes out the yellow pigments. And they did that to test whether UV reflectance matter more than white or yellow coloration. And the answer was yes, UV reflectance does matter a lot more than we give it credit. That is not just a white or yellow decision for mate choice. Yep.
That's a super cool talk. I love the
Thanks.
Really, really great stuff. But I want to take 1 second to thank Emma and you, Kai, for a great semester of Tuesday talks. So,